Welcome to the Division of Education, Innovation and Energy e-learning program. I am Ms. Jamilla Bacchus, your facilitator for CAPE Biology. And today we are looking at Unit 1 Biology on the topic of sexual reproduction in humans. This is in Module 3 of the CAPE syllabus. Now, when we hear the word sexual reproduction, a number of persons get very excited and they think this is what biology is all about. Good. Why is sexual reproduction important? Why aren't we all the same? Why aren't we all clones? What is the benefit to an organism of being sexually reproduced? Firstly, as we would have said in reproduction, we need to ensure that the species continue. But not just continue, we need to ensure that each and every person is a unique being. Unless, of course, you are a maternal twin, in which case you and your twin will look alike. But we all need to be unique in order for the species to be stronger and to be able to survive in spite of environmental conditions. So, because sexual reproduction brings together two sets of chromosomes. It brings together two different gametes to form this one unique individual that we are. So let's look at what happens during fertilization, what happens during implantation, and we will look a little bit at what happens during pregnancy. As we can see, the sperms, which we would have seen earlier on, are released by the males and they swim, they go through quite a journey, and eventually their focus, their aim, their target is the egg cell. Look at this diagram. This shows you the difference in the structure and the size of the egg and the sperm. The egg, this large round mass, and the sperm, these very tiny structures with their tails. And this is a beautiful picture which shows you the uniqueness of this whole merger of the male and the female, because eventually only one sperm can enter this egg. And I normally tell my students, whenever we feel that, my life is not of any value or anything, you just have to think of this fact. One sperm met with one egg to make you. There were many brothers and sisters potential that did not make it. Think about that. So, what are we going to look at today? We want to outline the steps involved in fertilization. We want to be able to describe implantation as it relates to reproduction, explain placental structure and function, and discuss the function of the amnion. We also want to be able to draw and label and annotate the placenta, draw, label, and annotate a fetus in early development in the uterus. Also to discuss maternal behavior on fetal development, and lastly, apply this knowledge to answer CAPE-type questions. So this is one such question that you can get, and we're just going to look at the question now. Again, take a picture of it, take a look at it, and in the end, we will come back to this question as we would have gotten the answer throughout the exercise. So again, this great race. So I want you to take a brief look at what is happening during this process.
During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, But here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida.
So here you have the head of the sperm, and inside of that you're seeing this area here where you have the acrosomal granule. And inside of that granule you have these enzymes which will be released. Remember, this sperm head would have been prepared for this precise function as it passed through, as it was released within the vagina and it passed through the uterus. It would have gone through capacitation where the tip of the sperm would have been prepared for this process. Now, you have to have this layer. The sperm has to pass through the zona pellucida, which is this clear jelly coat region here. And it has to pass through this layer to be able to get in contact with the membrane of the cell, the membrane of the egg cell. So this is the membrane of the egg cell, and the sperm has to be able to get there. Now, the first sperm that merges with the membrane of the egg cell, as you can see here, it will merge with the membrane of the egg cell, and so basically they become one. This now will cause the activation of the cortical granules to release their proteases, which will allow this layer to seal off this egg, and so no other sperm can enter. Some of them may be trapped within the zona pellucida and the outlying areas, but no other sperm should enter. You may ask the question, what if another sperm should reach at the same time? Well, if perchance that happens, then obviously this particular secondary oocyte will not develop. Okay, the body will reject that. So what happens normally, looking at what happens normally, one sperm will fertilize one secondary oocyte. So as this reaches here and the nucleus of this sperm is released into the cytoplasm of the secondary oocyte. Remember, this is still the secondary oocyte that is halted at metaphase 2. The last stage of meiosis will take place. So you go from metaphase 2 to anaphase 2, telophase 2, cytokinesis 2, and at the end you will form the nucleus that is now of the ovum, and you will also form the second polar body. So this will be the end of meiosis. The nucleus of this egg cell now is called the pronucleus, and the nucleus of the sperm will be the pronucleus. And what happens next is that they will both merge to form this new individual, a unique individual that is now the zygote. Good. So as fertilization occurs, you now have the merging of the two nuclei to form the nucleus of the zygote. This immediately will carry out mitosis, and you will go from the one nucleus and the one cell to two cells. But this cell now, these are diploid cells. They have the two sets of chromosomes. These will rapidly divide again and continue their mitosis and increase the number of cells. So you go from 2 to 4, from 4 to 8, and then from 8 to 16. Now this process where this development is taking place, where you are continuing to carry out mitosis and going from one stage to the next, this is called cleavage. Right? And what you have happening is increasing the number of cells, not yet specialized. And it is at this stage, if you do have to have stem cell research, that cells may be removed. Now, by about day 4, you have this structure known as the morula. And this is just a mass of undifferentiated cells. And then you form a structure known as the blastocyst. Now what you can see here is a ring of cells on the outside. And these cells are called the tropoblasts. Right? You have the trophoblast, and the cells which together make up this tissue in the middle here is called the inner cell mass. Good. So this blastocyst, this will be the last stage before implantation takes place. Notice on the outside of this structure, you still have the zona pellucida, this clear jelly-like structure. It's still on the outside of the blastocyst. So you have implantation, and this is the process where this developing embryo now attaches itself to the wall of the uterus. So implantation. Here we are seeing the development of this new organism, this new unique individual. You can see here, this is 7 days after implanting the blastocyst. What you can see here, now remember we said at the outer layer you have the trophoblast, and you will have some parts of this trophoblast develop to form these structures. They are called syncytotropoblasts, and these structures eventually form the chorionic villi. They are outgrowths, and they are specialized cells that will be able to invade deep within the endometrium to be able to allow the transfer of substances with the mother. Remember, this whole structure here is the endometrium. So this is the thickness of the endometrium. This is the mother's tissue. But this developing structure is eventually the fetus tissue. So what has to happen here? Now the mother's blood system would have been bringing blood, and these areas here are areas where you have blood vessels, And the mother's blood system will allow substances to flow into these blood vessels. So what you will have the chorionic villi forming that will be able to interface with these blood vessels, which will form sinuses and surround the chorionic villi. So these are the beginnings of the placenta. Here you are seeing a little more organization. Note too, you're seeing structures such as the yolk sac, which is the beginnings of the whole vascular system, the blood vessels that will come from the mother to the fetus. You also see something called the allantois, and the allantois is the structure which is chiefly responsible for getting rid of the waste, some of the gases and the other excretory substances the embryo would be producing. So at this stage, it's still an embryo. It's not yet a fetus. Remember, embryo is a mass of undifferentiated cells. A fetus, you will see differentiation, and therein would be the main difference between them. So what you have happening here, this structure has embedded itself within the structure. What is happening here is something called adhesion. It is now within the walls. The walls have surrounded it, and this structure is in the uterus, in the lining of the uterus, precisely what the lining was there for, and it is developing further. So here again, we can see what is taking place with the development. You have the outer structure, which is called the chorion, and in the part where they will eventually form the placenta, you see an increase in the surface area, an increase in the outgrowths, so that you can have greater exchange of substances. Again, in here, you are seeing the egg sac, and here you are seeing the allantois. Right? These structures are the forerunners to the blood vessels that will carry blood to and from the fetus. Here we are seeing the placenta, a diagram of the placenta. Note you're seeing the mother's tissue. You have the maternal artery and the maternal vein. And you're also seeing the fetal tissue. You're seeing the fetal artery and the fetal vein. What is happening here is that the mother's blood is flowing from her artery into this area here, this sinus, this space that is called the intervillous space. The intervillous space will simply be this came from the outgrowths of the capillaries we saw in the last slide, and all of the various capillaries would have joined up and formed this massive opening in which blood flowed. So it's just an open space. It's just a sinus that blood flowed into. And then we have the baby's tissue. You have the fetal vein. Note it is red in color. Red denotes high in oxygen, and note it is a fetal vein. So the fetal vein, also called the umbilical vein, is carrying oxygenated blood, but it is still representing the vein in that veins normally carry blood to the heart. The fetal vein is carrying blood to the baby's heart, but in this case, like the pulmonary vein we would have looked at sometime in Form 5, carrying oxygenated blood, right? So this is a deviation from the rule that normally veins carry deoxygenated blood. Here the fetal vein is carrying oxygenated blood. Note that you have one fetal vein and you have two fetal arteries. The arteries here represented in blue, they are carrying deoxygenated blood. So they are carrying blood from the fetus back to the mother. So what you have to note here is there is no direct movement of blood and the contents of blood between the mother and the fetus. Substances will get into this space and then through diffusion will move into the villi and move across. So these here would be the chorionic villus. One is a chorionic villus, and so the pack or the whole structure will be the chorionic villi. All of them collectively would be the chorionic villi. Here we see it a little closer. Note, as I said, it could be called the umbilical vein as well as the fetal vein. So you're seeing again the structure of them. Here you see nicely the maternal artery letting out blood into this space, right? This open space is like a pool that blood is being filled, is being filled up with blood. And these outgrowths would be the chorionic villi, which comes from the baby's tissue. What the structure of this villi, as you would know, anytime you have these enfolding and a villi structure, you would recall it helps to increase surface area. So the fact that you have this structure made up of all these infolds, it greatly increases the way in which substances can move across. It increases the amount of surface area for substances to move across. You have, as we said, this is the chorionic plate, and this is the basal plate where you will have the mother's tissue. So you have interface in here between the mother and the baby, and this here is a section of the placenta. What are the functions of the placenta? The placenta has many, many functions. It is the digestive system, exchanging of electrolytes, amino acids, carbohydrates, fatty acids, and vitamins. Respiratory system, exchanging of gases, carbon dioxide, oxygen, and you will also have carbon monoxide. For example, women who smoke will have an increased amount of carbon monoxide in their blood, and it will actually go across the placenta. The excretory subsystem, removal of waste. Endocrine system. Now, once a woman becomes pregnant, once fertilization occurs, she will begin to produce a number of hormones. One of the first hormones she will produce, as we recognized in the last video, was HCG, human chorionic gonadotropin. And this alerts the ovary that fertilization has occurred and implantation has occurred, because the HCG is only produced once the chorionic villi starts to be formed. So that ensures that the ovary continues to produce progesterone by the corpus luteum to maintain the lining of the uterus until the placenta takes over its role of producing the progesterone. So it has an endocrine system. It acts as a transient complex organ that secretes essential pregnancy hormone, estrogen, progesterone, HCG, and HCS. The immune system, it allows the transmission of maternal antibodies. Very, very important in protecting that baby, because that baby would not have immunity, it would not have long-term immunity. So this provides it with short-term immunity during this time. All right, it gives the baby passive immunity against any infectious diseases. I may add, the placenta also plays a very important role in anchoring the fetus within the uterus. If for some reason part of that placenta becomes detached, then the many functions that it carries out obviously will be affected. And so, you know, it's very important that when a woman becomes pregnant, that she maintains some relationship with her health professionals to keep a check on things like your weight and, you know, even your blood type and so on. It's very important that initially you do make some contact with a health professional once you become pregnant. So the placenta and the maintenance of the placenta health is very important in maintaining a healthy fetus going into delivery. This again is showing you the structure of the fetus. And note, this is a diagram you should be able to recognize to identify the various tissues. So we are seeing the chorion, which is the outermost layer, right? We are seeing the amnion. The amnion would be this tissue which surrounds, use a different color. So this tissue which surrounds the fetus, that tissue that is interfacing with the fetus is the amnion. So you have the amnion and then the chorion. The chorion is interfacing with the endometrium. Then you have the amniotic cavity. The amniotic cavity would be this space inside of here. Right? This space inside of here. And that is where the amniotic fluid will be. And we'll look in a little bit at the importance of that fluid. Of course, we will see also the umbilical cord. The umbilical cord, which would be this structure here. And it is inside of this umbilical cord that you have the umbilical artery and the umbilical vein. This is the fetus lifeline, the connection to its source of food, its source of, you know, gases, its removal of waste and so on. So the umbilical cord is very, very important in the development of this fetus. You have the allantois, as I said, this would be the forerunner to the structure that will be removing the waste, more or less the umbilical arteries. And then you have the yolk sac, as I said, would be the forerunner to the umbilical vein. And the chorionic villi in this area, which eventually would become the placenta. So you should be able to draw this diagram and label it and even annotate it going forward. You do not need, the syllabus doesn't require for us to go very in-depth in the gestation period to speak about what is happening during the trimesters of pregnancy. But for your own edification, you can do it. But the syllabus doesn't require that we go into the trimesters of pregnancy, neither birth.